Our next speaker is Andy Rominger, and he's going to talk about macroevolutionary signals of insular adaptive radiations.
All right. Well, thanks for sticking out to the end here. Hopefully the Caparines aren't calling too hard and we'll get through this together. So adaptive radiations are pretty cool. They often happen on isolated islands that form in a chronological fashion. And we really love, these are kind of textbook examples of understanding the drivers of diversification. So we'd really love to be able to understand how the rates of speciation, extinction, migration change through time and change as a function of interesting selective pressures that may be put on these organisms. So I work in Hawaii mostly. The oldest high island in Hawaii is Kauai, which formed about 5 million years ago. And we have this kind of classic story where that was the only island out there. A colonist arrived, diversified, something founded a new population on the next island, diversified, and keeps going down the island chain, diversifying. So it would be great if we could know, about extinctions on the oldest island and incipient species on the youngest island. The real big problem we have when all we have is a molecular phylogeny is that it can be really difficult to reliably estimate these rates. So in simulated studies where we know, say, the extinction rate is something, and we get a sampling distribution of what our inference method tells us the extinction rate is, oftentimes there's good agreement, but then there's this pesky peak at no extinction, which we know isn't true. When we look at real phylogenies, often it's even worse. And all of the density of the sampling distribution is right there at 0. So that's the worst case scenario, which is often quite common. And Quintal and Marshall pointed out that, pretty much phylogenies need the fossil record. That was their point, at least most of the way we do phylogenetics. So this is showing how the gamma statistic, which is a common summary statistic of phylogenies, depends on everything about a phylogeny except what you hope it depends on, which is extinction rate or slowdown in speciation. But there has been some success, and I don't want to ignore that and just say everything's terrible. So Helene Orlon has done some really good work taking coalescent approaches, applying them to phylogenies, and looking at time-varying speciation extinction rates. And you can see this is a comparison of her estimate and the real fossil record. So we have some ways of getting at the questions we might want to ask. And I was excited to know that this is all in our package now, so if anyone wants to use that, that's pretty cool. There's also Luis Valente's work, also in our package, that is specifically for islands, so that's kind of exciting too. But the goal of this model is to look more like a MacArthur-Wilson type thing. We have one island, immigration from the mainland, diversification on that one island. What we really want to look at is this evolving archipelago. And I'll point out, I'm always talking about Hawaii, but this could be applied to cichlid fish in rift lakes or anything where there's a age-structured landscape and a cool adaptive radiation that you want to look at in that landscape. So before I go into the modeling work we've done, I thought we might ought to get set on some vocabulary. So I'm going to be talking about cladogenic speciation, which is what I define and what others define as speciation that occurs within an island. There's also immigration in the model, as you can imagine, and then antigenic speciation is what happens potentially what happens post-immigration when a species form differentiates from its ancestor, not within the same island, but between islands. And of course, then there can be extinction both locally on an island and globally in the whole phylogeny. So, how are we actually going to fit those things to data? Well, likelihood, is the way to go. And it turns out that for birth-death processes, the sufficient statistic for the likelihood is just the vector of branching times in a phylogeny. So these red lines represent the branching time. And it kind of makes intuitive sense that that's all you need, because speciation adds a branching point in a phylogeny, and extinction effectively gets rid of it. So the two processes we care about leave a distinct signature in a phylogeny. Immigration is a little bit more ugly, which is why I went for the ugliest font and the ugliest color I could find. And it's the bane of my existence in this particular work. So it, oops. Well, there we go. It also adds a node or adds a branching event in the phylogeny of an island. So if a new immigrant arrives in an island that hasn't been there before, it adds a node in the phylogeny of that island. But immigration also means that the phylogenies between islands are not independent, so we can't just fit a model to each phylogeny independently and then somehow combine the inference of all those models. We have to jointly infer everything about all these differently independent or non-independent phylogenies. So that's been the tricky bit about this stuff. And that's the reason why I'm going to present a simulation study instead of pretty, likelihood surfaces and all that today. But if anyone has thoughts on this, it'd be great to talk about that. So what I wanted to do was really just use simulation to see if we're headed in the right direction, because if we simulate from known models and the summary statistics, basically the branching times that we get, are distinguishable between parameter sets, it means that we have an identifiable model. It means that once we work out the mathematics, we will be able to get meaningful parameter estimates. So I just picked kind of four, to me, intuitive models for diversification across an island chain. And on the x-axis is going to be the time since island formation. And then on the y are these different per capita, per species rates. So the solid blue is cladogenic speciation, dashed blue, antigenic speciation, yellow is extinction, that lovely teal turquoise is immigration. And what we're going to be looking at is, just for fun, we'll look at the gamma statistic and also branching times. And then we'll also look at species richness, just because that's something that we often care about, right? Adaptive radiations are adaptive radiations because there's so many cool species that arise out of them. So I'll just kind of get everything on here and then point out interesting things that are going on. So I think one of the first things that's kind of interesting is that across all these really quite different model scenarios, the thing that predicts the gamma statistic most consistently is actually inter-island immigration. So a gamma statistic is thought to be an indication of speciation slowdown for the present, but what it seems like is it's a measure of panpictia of species between pseudo-independent landscapes. So that's a result that really surprised me. I don't know quite what to make of that. Another thing, if you care about island biogeography at all, you probably have heard of these overshoot hypotheses, and there's different hypothesized mechanisms behind them, but basically in islands that have an age structure, we always see, or tend to see, peak diversity on middle-aged islands, and then less diversity on the young islands and on the older islands. So the thing that comes out of this simulation work is that that kind of peak in diversity can come from a lot of different possible parameterizations, including just flat rates. So the shaded area is the 95% quantile region. So you can see that in some simulations you actually had a pretty strong peak in diversity at middle-aged islands just when there's absolutely no rate variation at all. You also get it from two pretty different trajectories of speciation extinction across island age. And then the most important part statistically is that you get different patterns of branch time, vectors of branching times depending on what the model is that was used to simulate the data. So that's just indicating that this will be an identifiable model once we get it all up and running. And once we get it all up and running, we can do cool things. So I think one obvious thing would be to look at, actually make a hierarchical model where the rate parameters are themselves functions of other things that we care about in the environment. Island size, topographic complexity of islands, interacting species on the islands. But the other thing that we're really interested in my group is understanding how evolutionary history has shaped contemporary ecology. And so much of this work, so much of this research done on this question has been kind of very, not just so stories, but we have no way of really quantifying history. So we're left with talking about the history of a region and saying, hey, and look at this diversity pattern, it must relate to the history somehow. If we can quantify evolutionary history with maybe some model like this, then we can relate whatever it is about the history that we're interested in to things like shape of species abundance distributions. And we care about things like that because they're really important for thinking about extinction, thinking about invasion, things we might be concerned with from a conservation perspective. And I think with that, I'll wrap it up. Thanks to people who talked about this, and thanks to people who funded this.